I am Mr. Fizzle. I'm actually married to Mrs. Fizzle. She wasn't feeling well today, so I took over for her.
So you're teaching us today. You're taking over?
Yes, that is correct.
Oh, phew. She usually takes us on these crazy adventures, and then people end up getting hurt, people get lost, and, you know, she's a little crazy, but thank God you're taking over. But why hasn't she talked about you before?
Oh, I actually got out of the mental hospital recently. I was trying to shrink people so that they could enter cells, but then some of them didn't like it very much.
Oh, brother. So you're going to be teaching us today?
Yes, that is correct. So, I heard that she was teaching you guys about meiosis, but didn't get to do that much.
Yeah, I don't really understand it.
I was hoping you'd say that. To the magic school bus!
No!
Since I am really bad at remembering things, I thought that I would just show you what meiosis was, and I'll explain you each step. We are now passing the cell membrane to see where this whole process starts.
Okay, I think I remember reading something about prophase starting meiosis.
Yes, that is correct. You see that right over there? DNA is coiling tightly into chromosomes.
Oh, and are those spindle fibers that are appearing, like in mitosis?
Yes, they are in fact. This is also the part where the nucleolus and the nuclear envelope disassembled. And right over there, each chromosome is lining up next to its homologous. Each cute little pair is called a tetrad. This does not occur in mitosis. They call this synapses.
I think I kind of remember something about tetrads. In the chromosomes of homologous chromatids are aligned lengthwise so that the genes on the chromosomes are adjacent to the corresponding genes on the other chromosome?
Yeah, so anyway, during synapses, the chromatids twist around one another. Some portions of the chromatids may break off and attach to adjacent chromatids. This is called crossing over. This is so genetic material can be exchanged between maternal and paternal chromosomes.
Oh, doesn't genetic recombination result because there's a new mixture of genetic material?
Shut up, please.
Sorry.
Anyways, we are now on our next phase.
Metaphase.
Metaphase. Tetrads line up randomly along the midline of the dividing cell. It is random, but with respect to the poles of the cells.
Did you by any chance have a violent past?
Ever wonder how Michael Jackson really died?
Shutting up.
At this point, spindle fibers from one of the poles attach to the other homologous of the pair. I think next is anaphase.
I think so. Isn't that...
Excuse me?
Shutting up.
Since you're so anxious, I'll get through with this quickly. Some kids just need Ritalin.
Hey!
So, anyways, anaphase is just when each homologous chromosome moves to an opposite pole of the dividing cell. That random separation is actually called independent assortment. This random assortment actually creates genetic variation.
I get that part, but I really need help with this next part. I think it's telophase and cytokinesis?
Telophase is just when chromosomes reach the opposite ends of the cell. This is when cytokinesis begins.
That's it?
No, it's not it. Can you start to see that the new cells contain a haploid number of chromosomes?
Yeah, I could see right... That was a rhetorical question, right?
Everything I just described to you is meiosis, but it's known as meiosis 1.
There's more... Oh no...
Oh yes. In meiosis 1, the original cell produces two new cells. Each contains a chromosome from the homologous pair. However, the new cell only contains half the number of chromosomes the original cell did. But
each new cell contains two copies because the original cell copy is DNA. I'm going to shut up now.
Now, since we're running out of time, I'm going to quickly explain meiosis. This is what occurs in each new cell after meiosis 1. You and your stupid questions. In prophase 2, spindle fibers begin to move the chromosomes toward the midline of the dividing cell. In metaphase 2, the chromosomes move to the midline of the dividing cell with each chromatid facing opposite poles of the dividing cell.
So then would anaphase be when the chromatids separate and move to the opposite ends of the cell?
Precisely.
Yay! What are the next steps?
In telophase 2, a nuclear membrane forms around the chromosomes in each of the 4 new cells. Cytokinesis 2 can now occur, resulting in 4 new cells. Each contain half the original cell's number of chromosomes.
Thank you! I learned so much. And I remember that meiosis was the primary event in the formation of
Gametes? How is this?
You see, meiosis only occurs in cells that produce gametes within the reproductive organs. The location varies in organisms.
So it would take place in the testes of men and the ovaries of women?
Exactly.
Oh no, it's 4:09. School ends in 1 minute.
Let's go.
Seatbelts, everyone! Please let this be a normal field trip. With a friend? No way!
Cruising on down Main Street, you're relaxing, feeling good. Next thing that you know, you see it. The first in my neighborhood, surfing on the sound wave, swinging through the stars.